So let's make sure you're in the right session. Welcome to the multi-tissue integration breakout session. If that's not the session you were hoping to take part, well, I will invite you to switch rooms. Maz and Nifa is unwell, so we have Kerstin Meyer who's joining us. Just to kind of get a feel for the rooms, the three of us would love to hear, by show of hands, who here is attending this meeting for the first time? It's your first HCA meeting. Oh, that's exciting. This is like a good 30% of the room. Okay, some of you have yes, hello, yeah. Some of you have multiple stickers, which tells me you're part of multiple bionetworks. So here we're talking about multiple tissue integration, which, as you've heard, we're going through different waves of integration. Right now we're focusing on individual bionetwork integration. But a lot of these cells are shared across tissues, and even though we won't get there before wave 3 and perhaps wave 4, we already have to start thinking about these frameworks ahead of time because, there's a lot of data management, methods development that needs to be tied to it. So I'm really curious to hear the breakdown of the room. And there's 18 biological network. I will go through 18 biological network. Just by show of hands, who's an adipose fan? Uh-oh. Super underrepresented. Okay. Well, adipose, super important. They're everywhere. I have quite a few cells myself. So hopefully we can tie them in later. What about breast? Thank you for being here. We have development. Kerstin, you're there. Okay. Oh, yes. Okay, development we have two. Eye. Okay, genetic diversity. Shyam. Okay. Oh, yeah, we have some representation. Gut. Thank you. Heart. Okay, we have representation. Immune. Hey, my friends. Kidney. I'm glad we have representation for almost every organ. Liver. Thank you. Lung. Muscular skeletal. Okay. Nervous system, brain. I saw Ed. Hey, Ed. Oral, cranial, facial. Organoids. Think pancreas. Reproduction. Skin. Hello. Okay, good. Well, it's actually pretty good representation. And so we posted our questions on the website, and we thought what we really hope to do is not so much the three of us answering these questions, but we really want to hear your thoughts. We have amazing scribes that are here today. We're going to be taking notes of everyone's thoughts. It will be really key in defining our next steps in planning this multi-tissue integration where everyone should have a say in them. So we've broken them in terms of the big picture questions, right? So first, the question is there, but I'm going to read out loud for those people that are online. To all of you, what does an integrated atlas look like within an organ system and across system? What data modality should it include, and what are priorities of data in areas? I mean, if your answer is all data modalities, I'll be like, sure. But given that we don't have all the money in the world, what do you think should be a priority in time if we wanted to integrate it in wave 3? And tied to this, you can decide to not answer this question. You can answer the second question. What are good case scenarios for you about cross-tissue analysis? And there's a reason why they had invited Shyam, who represented genetic diversity bionetwork, Kerstin, who's here, sitting in from us, who represents the development atlas, and me, who's presenting the immune atlas. These are kind of clear cases, but we want to hear more specifically. I saw a hand go. Yes, I don't know where the runners. We have. Wow, so close. Yes.
Hi.
Can you please introduce yourself?
Yeah. Hi, I'm Hari. I work on breast cancer. I use the term cancer here. That's why my interest in cross-tissue epithelial cell types.
Oh, that's a good one.
So, particularly the cancer community is more interested in knowing what's the difference, whether there is a lineage plasticity which cancer cells acquires, whereas the breast epithelial cells acquiring the lung epithelial phenotype so that they can metastasize to lung. So those are the things I'm interested in knowing.
That's actually a really good one to put on the radar because we were not discussing it. That's great. With another hand right up there.
Thank you. Hi, Sophia George. I am actually also interested in what Harry said, but from the perspective of normal tissues, so breast, fallopian tube, and endometrium. These cells are hormonal responsive. They express estrogen receptor, progesterone receptor. When you think about cancer, the really aggressive types, they're genomically very similar. So I would like to see how similar they are in a normal setting.
Okay, so hormone-producing cells across the human body, endocrine system. Any other thoughts? Shyam.
Actually, you're right, Chloe. Even I was thinking mainly of immune cells, but if you think about it, fibroblasts everywhere, and they have crazy diversity within one tissue itself. So the question is it even more crazy when you look at lots of tissues, or do you see something repeating?
Yeah, we heard colleagues this morning talking about vasculature, which is another one who made us reflect on the fact that we don't really have a vasculature bionetwork, but these cells are very critical, something to reflect on. That's another good one. Kerstin, any thoughts?
Yeah, I mean, maybe I'm actually sort of putting a plug here for the developmental network, because actually what we have the opportunity to do with the developing embryos is actually profile across all the different organs at the same time. And so actually we have an opportunity to get organ-specific data with minimal batch effect between different organs, because at the end of the day, they're all from the same individual, taken at the same time. And there will be like soup issues because you're taking potentially different fraction of the embryo, but still that's a lot less of a batch effect than you would normally expect across the many different organs that we take from adults. So, maybe even the developmental atlas can sort of serve as a bridging tool in some ways.
And maybe some of our follow-up questions, and I see Ed has the mic, so I'll get to you in a sec. But, I'm so sorry. Yes, I'll get to both of you. But just to kind of follow up on this, and maybe we table that question, but just to flag it, it's so true. That's one of the values of the development atlas, but you have that dynamic, the biology is changes constantly.
Yeah, and that obviously adds another whole layer.
It's a whole different layer, right? So in your case, you got the layer of cross tissue, but the time window where that biology is fixed is probably very small, which makes it very hard to think about how do you integrate, because you need to think about the temporal aspect of it. I mean, I think it's a question that's coming up. So let's table this for a second. And I don't know who was up first. Ed.
Yeah, I just wanted to say I think there are many, many cases where you'd like to have whole body analysis. Imagine genetics of disease, where you'd like to understand how a risk allele affects cell types in different organs. Or from an industry perspective of wanting to be able to predict off-target effects for drugs that hit a particular molecule. So, I mean, you'd really love to have this whole body cell and molecular atlas to be able to look at those things.
So you're basically pushing to make sure that we try to integrate genetic data systematically with our atlas. No, I'm supportive of it, just making sure we write this down. So
Yes.
Okay. Yeah, I completely agree. I mean, there's rich data sets of genetic association studies out there where we're missing the whole cellular context of it. So there's a huge opportunity to start bridging between these two fields. Yeah, couldn't agree more. Yeah. Kerstin.
My comment was actually identical to Ed's, which is that I was going to say if we look at the GTEx resource, which is a multi-tissue across individuals, huge number of uses of tissue specificity of effects, and people want to know cellular specificity. So this gets us to integrating the distinction between integrating atlases across organs that are from different individuals versus the same individual. And I think you can answer that first question by integrating even if they're from different tissues and individuals. But the genetic one is really key, and we've been interested in genetic effects that really manifest very specifically in a tissue or organ type in a specific cell type. And that cell type may be shared in another tissue, but the effect is manifest. So it's a really critical to have this integration to understand that, I think.
So Shyam, you have a lot of supporters of your bionetwork. I think I couldn't agree more with Kerstin and Ed. I think one of the challenge is, aside from the genetic diversity bionetwork, who proactively actually collects this type of data, most other, and perhaps the brain does. Maybe you do, right? You get the genetics. A lot of the other bionetworks don't systematically do it in terms of where the money is put in genotyping. So, to make genetic worthwhile, you also need to have the cohort size to some extent to be able to capture the full breadth of genetic variant frequency. And I think it's important to reflect on this as a community. If it's, I mean, I know maybe you want to expand on the scope before I pass it to Malta, like of what you're focusing on and which tissue your roadmap has. And if we should encourage, as part of the next wave of Atlas building, for people to really embrace genotyping their subject. Because at the end of the day, if you have limited resources, you need to decide, am I going to do more spatial? Am I going to do more genotyping? Where do you put your money is the question.
Yeah, thanks very much, Chloe. So I'll start with a more general goal and ambition. There's one thing to say, it sounds almost pious, right? That, we want to represent everyone and so on, but it's true, we do. But if that does not inspire you, maybe what will inspire you is that scientifically, the thing that you think is true biologically in, say, your population may not be true in a different population. So you think you're studying human biology, but human biology is very contingent on which humans you're looking at. And you may be actually misled and publishing something that's very, very narrowly specific to your population. So that's certainly what Hari is finding in even normal breast tissue, and we're seeing in PBMCs and so on. That even simple things like what is the male-female difference is not, if you study the male-female difference in one population, you think that you've studied humanity, you have not. The male-female difference in a different population turns out to be different, startlingly so, right? Meaning the whole effect, it's not that the effect has gone from X to 1.1X, the effect is not even there in a different population, but the effect is in the opposite direction in a different population, right? For all those reasons, I think if we look back at our research 10 years later, we'll say, why did we not genotype everyone when we did single-cell sequencing? Because now 10 years later, we know that almost everything is contingent on your genotype or your environment or something like that, right? So this is really a push for collect as much metadata as possible on your individuals. Even if you feel, let's say your cohort is not big enough for QTL analysis, that's okay. Your cohort may still be big enough to say that men and women are different in this way, or males and females are different in this way, or say European versus African versus Asian, to use very crude terms which we don't want to use anymore, are different. And the genotype data can tell you that, right? In hindsight, you'll say, okay, I really needed that to really interpret my results. Then coming back to the other question, what is the GDN doing? We're starting very small. We're just looking at PBMCs because we have large enough cohorts where we can fairly, with some level of confidence, say, gender or sex, geography, ancestry, genetics are correlated with this expression trait. To say that, you need a reasonable size cohort, and that's why we're starting with PBMCs. There are other tissues, actually, where you have reasonable size cohorts, you can do this analysis. But we have to start somewhere, and blood is the easiest place to start.
So, clear call about genetics. Malta had a question, and I'm going to pass it to you. Yeah. Or comment, I should say comment.
Yeah, I just want to add also what I think is really exciting about cross-tissue analysis is kind of cell migration, maybe across tissues. And something that we heard this morning also is this kind of mitochondrial DNA enabling tracing, lineage tracing in humans. And I wonder, this is very new, right, of course, but if we start going cross tissues and we are able to do this, especially from multiple samples from the same individual, that would be really exciting, I think. To be able to really kind of track immune cell recruitment, for example.
Yeah, you want to answer back to this, right? Because then I want to, there's two other comments.
I was just going to comment that in the Human Developmental Cell Atlas, we actually have been able, and Maz's lab has really led the way here, been able to sample multiple different organs from the same individuals. So, and we are set up to do genotyping on all the different individuals that we're collecting samples from. So I think that, again, will be an opportunity to really start to go quite broad. And, I think from a practical point of view, it made sense to start all the different atlases for one organ at a time to be manageable. But again, also for the Teichmann team, I can say that we are retrospectively genotyping all of our individuals. So.
Just to close, that's helpful, to close on Mata's comment before we switch, I think that's an important point you brought up and that we need to flag. It's not just about discrete cellular identity, there's a dynamics aspect of it, and there are indeed a few efforts that are autopsy-based where they're collecting multiple tissue that can serve as a strong scaffold to start answering these questions. And then for the data sets where we don't have cross-tissue, we can try to see how we can start adding some resolution to it. I think that's an excellent point. Thank you. Yeah. Yes.
I think Shyam mentioned this, the sex-specific difference we should not be ignoring as we move forward with the recent flurry of papers showing that the Y chromosome is not a desert. It produces at least 18 transcripts. Some of them are epigenetic regulators, which are differentially affected in cancer. We have started creating the reagents. We just made the single-cell breast atlas of the men as well as women. We are superimposing on that, and we are creating the cell lines from men breast epithelial cell lines and the women to compare. So I think moving forward, we need to be we need to make sure that we consider the sex not just based on the hormone, it is the Y chromosome as well.
That's great. Matthias.
Yeah. And I think heading forward with multi-strategy of cell type classification in each organ system, these cells behave similar under similar states across different organs, and the solution developed in the lung might work for another epithelial or mesenchymal organ as well. So the discussions, how do we define resting, cycling, adaptive, maladaptive epithelial cells? Now, these characteristics are similar across organ and might help you to also resolve some of the nomenclature discussions we had yesterday afternoon.
That's a really interesting concept. It's a good point. Trying to kind of rethink how we define, right, these different cellular functions. Okay, so what I'm hearing so far about, just to summarize this point, because I want to bring up the next questions, and you can still go back and answer these questions. So we knew already coming in about immune and genetic diversity and development. We heard about epithelial cells. We heard about hormone-producing cells. I pointed out vasculature. We heard about migrating cells and just not forgetting about the dynamics of these cells. We don't have adipose representative, but I should say adipose tissue is probably pretty relevant in terms of cross tissue. And the reason we're bringing this out, and some of it you may be thinking like, this is obvious. Shyam pointed out fibroblasts, is because we need to prioritize. We need to prioritize efforts. Where do we start? Where does that have the biggest impact? Because as we go in and integrate, do minimal viable product for each of the bionetwork, then for some of these common themes, we can start pulling out some of these key cells and subsets and start the next wave. And so that's why we're asking these questions. If you think of any other themes that we missed, and this is of course in the context of health, do bring it up, and it's never too late to reach out to us afterwards. In terms of data modalities, I mostly heard about DNA, so great publicity for everyone's genotyping. I think it's important. Just saying the obvious, a lot of the minimal viable product are being done out of RNA because it's the modality that has been scalable for a while now, and so a lot of the data set available right now are in the RNA space, some with proteins, some with additional modalities. A lot of people are now focusing on spatial, which is really key. Where do you put your money? Where do you think the community should put their money? More cells, more individuals with the same modalities? I'm sure Shyam will vote for that because that's better for diversity. More of the same? More sampling of a tissue to make sure that we're not keep on sampling a small piece. I'm sure Ed had some thoughts about this because Brian has been leading the way in terms of being very thorough. So more of the same sampling, more individual of the same modality, or we switch modalities completely. Any thoughts on that? There's some thoughts, and then I'll get to you.
Hi, I'm Erica from Stanford. Isn't there a third option, which is to add in more disease states?
Yes, that is an option. Yeah. Do you say we know normal? Should we? So, okay, I'm gonna switch cap and go from moderator to Chloe speaking for herself. I actually, as someone who cares about immune cells, I don't think we can define what's healthy immune cells without knowing disease. because it helps to define the boundaries of immune because of the dynamics of it, but I don't know if that applies to all. You can see Ed is itching of saying a comment. He's right there. And then Kerstin has a... so yes, disease could be one. We could say, but like, which modality, same modality, but on the disease. Yeah, okay, we wrote this down. Yes, Ed.
I mean, I think except for immune system, the concept of an atlas is coverage of the organ to try to understand the component cell types and their local tissue microarchitecture. So, you know, and brain is the most complex because basically every part of the brain has its own unique cytoarchitecture. But I think it still would be useful to stick with that basic premise for most systems, that you're trying to create an atlas, and you're trying to get that complete coverage, and to create enough representation that you could interpret where your samples came from if it was a biopsy or something like that. You know, even fairly repeating structure organs have gradients and things across them that dictate their positionality. So, you know, with that in mind, the transcriptomics still leads the way on defining the types. Spatial is needed to define the tissue architecture, and epigenetics is useful to understand the gene regulation, and there are other downstream practical things you can get from that. Today you would do it with multiome probably. So my recommendation still for most organs, and maybe immune system is not that, is to make sure you cover that. Create an atlas for each organ and do those basic things which give you that architecture.
I agree. So just keep the mic for a sec. That's very helpful insights, and I know the Brain Initiative has been thinking very thoroughly about this. So you go deep into a tissue from a single individual. How do you deal with the diversity of how representative your atlas is? Because it's very hard to go deep like you're doing, but I agree, this is how you do a proper chart and make sure it's representative of a bigger cohort.
So I can really only speak for brain, and for the most part, brain has been a super small N endeavor. You know, most of these studies are N of 3 to 6 maybe. But what's pretty clear is that there is an architecture that you can define the commonality across individuals, even from a super small number of individuals. And then when we expand the number of individuals, we can find those cells, those cell types in every other individual. And so that actually seems to work for understanding the basics. Then when you want to understand diversity, it's a whole different question, you know, because there are often more subtle effects that you need big numbers to go after. So I think that there are actually kind of two separate approaches. You can achieve the atlas in a small N effectively, but of course it will get embellished over time as you see whether there's more to be learned.
So what I'm hearing to summarize is, because I don't want to put words in your mouth, maybe I am, small N, but deep characterization of that small N in terms of thorough sampling of different parts of the tissue, but also measurement through different modalities to get a full picture of that small N, which hopefully allows you to derive broad principles of your atlas of your organ.
That's my recommendation as a starting point.
Yeah, I agree. I just want to make sure we summarize it correctly. Yeah.
Yeah, I was going to comment. Effectively, I very much agree with you that, you know, the transcriptomics has been hugely successful in determining cell types. We've had, as always, like the second pillar of the human cell atlas, the spatial, right away. But I think the one aspect where that sort of paradigm might change is if we actually truly develop spatial technology that can give you unbiased whole nuclei in space. Like we talked about, you know, earlier on, we heard about the tag-seq. If that actually works, then we don't need to do single-cell sequencing anymore, right? Because why not get the whole transcriptome and you get the spatial information on top of that? So, yeah, I think with those technologies coming along, I think there might be a shift with a lot of us moving to more spatial. But as of today, I completely agree with you.
I think that comes with the challenge of having, you know, when we do also your hand, when you do integration, you need to start now. So you need to go with what we have now. And what we have now ready to go is not the new data from the new, you know, the new methods that still need proper benchmark, and they have not been used at scale enough for us to do something. But I'm with you, things will change. I think Malta had a question, and then I'll come to you. Why should I have left for getting hands? Oh, I'm so sorry. Okay, she's been waiting for a long time. I am so terribly sorry. I'm gonna stand straight so I can see everything.
I just wanted to add to the conversation about going deep and
Oh,
few.
please. Yeah.
So when I think about the tissues that I study, predominantly in women, then we think, well, how few? Because we have premenopausal, postmenopausal, and that's part of the normal spectrum. Not talking about disease yet. And so diversity also means like states of
Like the
our
demographics.
existence. And I would assume it's the same for the brain too, right, in terms of fluctuations in people's responses to the environmental stresses and different environments that they live in.
Yeah, and also we didn't touch on this. We went from development fetal, we skipped pediatric, but there's also like the whole pediatric piece, which is not mini adult. And so, yeah, it's its own category. There were hands here.
Yeah,
Do
here.
you mind, Malta, and then you? Yeah, go ahead. Please introduce, go ahead.
Yeah, all right. Just go in the direction that Cheyenne raised before about the diversity and so on. One thing that I worry about is annotation. It's not always easy to see this difference between the different populations. We don't have good ways, especially when we have admixed populations but that go in the direction that if we have few individuals and you characterize them very deeply, then we might perhaps find some cell types that are specific for that population. I mean, there are some. I have a bone in my feet that not all people have. So there are these kind of quirks and particular anatomical things that some communities might have. And then if you analyze in depth few individuals from a common origin, you will perhaps get some false positives if you see it at the level of species, right? So I guess what I'm trying to bring to the discussion is that if we're trying to build the average or the core map of cells across humans, like the cell identities, they're shared by every adult human in some way, no matter where, no matter which origin, or are we trying to build an expanded version of this map that includes all the different tweaks that you have? So going in the direction, you have postmenopausal women, you have, I don't know, many different aspects of that. So where do we want to go? Do we want to go all, everywhere at the same time at once? Or do we want to go to the core kind of features of human beings in general?
I think that's an important, and there are a lot of hands being raised. Matt, is it related to this? The question, yeah. And then, because he's been waiting for a long time, and then, I know you have, but you're a moderator.
I can wait.
Need to let them speak. And then at the back. Just like quick thought though, like this, it's important for everyone to remember that this is a living effort, right? Like it's not, none of it is going to be final, at least certainly not in the coming years. Everything needs to be time-stamped so that we know that at this particular time, this is what we know, this is what we have. And we're likely going to get it wrong in retrospect because as we add more samples, it's going to become more and more precise. And we'll find the funny bone you have, eventually. Thank you for sharing this. But yeah, but it was a really good example to illustrate your point. Like, I think it's important to remember that none of this is final. It's really a living project. That's why it's really important to be able to track timestamp. That's why it's so important to have cell ontology piece, which I know you work on, and so forth, so we can track the evolution of all of this.
Thanks. I really like your point, Diego, as well. I think there's another aspect about, I don't think there's a right answer in the deep dive versus breadth, but another aspect that I'm thinking about is that I don't think there's a perfect protocol. And so if we have one really good, really well characterized protocol, there's always going to be something that's missing. And even if we go to multiple, different modalities, I think there is kind of multiple views of a healthy organ, which you'll get part of from different types of doing this experiment. And of course, I'm also, biased as a computational person. I want to train models, and I need a lot of batches of data to have a generalizable model I can train. And there's also that, I think, for uses downstream.
Right. So I think you're right. You're right that in the first, to bring context to Malta's excellent point, love the first wave. At the back, and then she had, sorry, I took the mic away from her, and then you, but first at the back. Just to Malta's point, just to bring context, a lot of the first waves I've been focusing on one type of technologies, just for sake of working out the quirks of the frameworks, which is 10x genomics. But there's a lot of data sets out there that each have their strength. We had this whole discussion over the immune bionetwork over lunch about how we were excluding neutrophils systematically. That's a good example. So to Malta's point, we should not be ignoring these other technologies. It brings a lot of richness. That's a good point. At the back, and then here, and then there. That's at the back.
Yeah, I don't know, I just think it's a little dangerous to think of genetic diversity as not part of the core of what we should be studying, but as like an add-on. I don't know, just in
I'm sure
response to
Shyam
that.
has some big thoughts about this. Yes, you can answer that.
Yeah, thank you so much. I think we should always look at it from the perspective of if we come back in our grown-up selves and come back and look at the decisions we made today, will we feel we made a mistake, right? So I think so I see Ed's point that maybe the brain is the most, in a sense, protected from the outside environment part of our body, and usually the parts of our body that are protected from the outside environment, they evolve more slowly. And so maybe that's a biological argument to say, okay, the brain should be more similar across people, maybe. But our skin is visibly different in different parts of the world, and skin is a part of the body that faces the outside world. It evolves fast. Our gut probably evolves fast. Embryos, early stage embryos evolve very, very rapidly across species. And so it won't be surprising if they diverge quite a lot across different populations as well. So I think the answer may be different for different parts of the body. I'm going to stick my neck out and make a bet that there will be organs we have where even the basic cell types are different in different parts of the world. In a noticeable way, not spectacularly different, like their fibroblasts are missing from this person, or not like that, right? But this fibroblast subtype is barely present in this population. This is hugely abundant in that population, so on. So I do feel that it has to, for many organs, we'll realize later that we should have really done it across the world or across many populations right at the start. And that's my guess. I don't think there's a lot of evidence behind it.
Yeah. Well, that's the whole premise of the bionetwork. Yes, we have a question here.
Hi. But then a question is kind of a comment, which it's okay, that summarizes a bit what we've said. And I definitely agree with Ed. I work on the heart and the vasculature, and understanding the difference in the anatomy within the organ across the body is fundamental. But to define the core, we need to include the genetics. We need to include male and females and expand the numbers. Otherwise, it's a core representative of a very small population, so we risk to skew it. I mean, we know that across ages, if you look at a 20 years old to a 70 years old, a male in female, things change a lot. The hormones change. Yeah. If you think the blood pressure changes, so that will affect the brain, to the heart, to everything else. So I really think we need to keep going with numbers and embrace all the diversity to define the core, which is across everyone, and then to define the spectrum that represents the more specific groups.
So if you don't mind keeping the mic for a second, I want to do a follow-up. I think it's an excellent point in talking about the heart for a moment. Of course, there's different types of disease types with different age and demographics, and so if you're given, making it up, I don't have $100 million to give, but if I have $100 million to give you, do you do a lot of the same modalities? I don't know, I'm making it up, RNA, but across all different parts of the heart in a lot of individuals? Or do you do different modalities for a smaller cohort? Like how there's no perfect answer, but the balance between increasing the numbers versus diversifying the type of measurements.
My gut feeling would be to increase the numbers first.
With the same modalities
with
first to kind of establish a foundation.
1, 2, 3 modalities rather than keep skipping.
But like thorough sampling across the heart, because there's different functions with different parts of the heart. There's a question he's been waiting for a long time on the left, and then you can, on the left, then Christine, and then Sergio, yes.
Okay, good afternoon, my name is Javier. I'm in the Schwartz lab here in the Princess Margaret. I just wanted kind of like to emphasize the necessity of having a precise objective, and what I mean by that is like, okay, so this is an atlas, so people have discussed about the differences in, do we need a pediatric atlas or something like that? So we could define precise guidelines like, okay, let's aim for having for every age, 1 year, 2 years, 3 years, 5 years, up to 100, 110 classifications of cells for those stages of age. But that, of course, would not be sufficient, right? How do you characterize differences in race, for example? So we could use the single nucleotide polymorphisms, but still that would not be sufficient, right? Because every organ would require, you need to specify some resolution, right? So we should define it precisely. Okay, for the heart, how precise we want to be, right? And either micrometers, or we already have an atlas for the heart, right? So we could define for the heart, we want to characterize, or at least take so many cells for every known region of the heart, or at least well-characterized region of the heart, and the same for the thymus, and the same for the brain, and so on, right? So even though it seems like a titanic endeavor, but if we write those guidelines and then we just work towards it, and then somebody will say, I have cells for a 3-year-old from Indonesia. Okay, perfect, right? And that we can just fill up this mega huge database.
Yeah, I think, thank you for your input. There are roadmaps that were written by every bionetwork in their community to try to exactly get to there in terms of where they should be prioritizing. And that too is, of course, a living document, because with emerging technologies and proceeding with your first integrations, you get to figure out the gaps. We may think we know the gaps. We may think we know which modalities work best. That's when you first write your roadmap, and then you realize that you need to update it. So I want to point out that I know it's probably hard, and others they have written, and everybody has written a version of their roadmap. I think what we haven't spent a lot of time doing yet, this is a topic of this session, is how do we align between biological network in terms of our roadmap? And that's perhaps something we need to focus on a bit more so that we can prepare properly for this tissue integration. So that's a good point. She's been waiting for a long time.
I just wanted to say, I guess, in follow-up to that, Ed's point about the deep, which we've been talking about, is really the right way to go, and that's where we started with the organ networks. But I think without sort of branching into a million different assays, we've got a range of assays now that we have working, and we keep discussing it, but one of the angles we're missing now is in understanding some of that diversity within an organ, taking a couple of key points and getting the diversity or the depth angle across. And we haven't done it. With or without genotypes, we haven't done it. With genotypes, is a no-brainer at this point, so we can add that information in. But also this session is about integrating across the different networks and tissues. And you asked which network we're in, and I didn't stick my hand up other than the pediatric one, because I'm across tissue. So suggesting that we should get some multi-cross tissue within individual is really powerful, and just the same types of data we're doing, because it controls, you can look at that diversity and integrate within individuals, and we don't have that represented.
That's great. That's great input, Kerstin. I think we need to think about this framework. This is really good input. For example, for immune, the way immune is structured is by tiers. So tier 1 is based on immune-specific tissues, and nobody else is profiling. So of course, blood, bone marrow, cord blood, thymus, lymph nodes, and even within that, we created sub-tiers. And then there's the mucosal barrier options. This is our tier 2, lung, gut, skin. We're waiting for our counterparts to proceed with their integration so that we can pull out the resident memory cells and then work with each of them to integrate them. And then tier 3 is all the rest. And we started with circulating cells, because if we cannot phenotype our circulating cells very well, it's going to be hard to map their resident memory cells. So in that sense, the immune bionetwork plays a key role in bringing these different counterparts together, the same way adipose, which is very tissue cell type centric, plays a different role. But it is true that for other cells, like the fibroblasts and I guess vasculature may fit with heart, actually, now that I think about it. Sorry about that. It very much fits with the heart. But you're right, Christine, with Malta's point about how it connects and the dynamics of it, maybe it's a different structure than we need to think about, so we can brainstorm more. Where's Sergio? I remember you raising your hand.
Yeah, I just want to send the ball to Shyam and all the whole audience. We are discussing here that we are different, but we aim for a human cell atlas. So why, in the same way that we broke the human into different organs, why don't we break the human community into different, I don't know, groups of people, and then we do this, focus in this group of people as Ed said before.
It's a good social experiment. We're going to put you in charge. No, it's a good idea. It's a good thought. Yeah, I don't know. Yeah. I think we spend a lot of time each of us just focusing on our tissue, and it's the next phase is not trivial. I think Malta has some thoughts. Malta, this whole next series of questions was for you. You almost need to be on stage. I wanted to hear also from the next series of questions. I'll let you speak, but because we have 20 minutes to go, I do want to open up to all these other questions. Before I give you the mic, Malta, keep the mic. The other challenges about, so first we're like broad question. We wanted to hear from you about where should we focus. We're not taking decision. We were going to summarize and report and give the opportunity for everybody to give feedback. It will help all of us as a community to set our priorities. But there's a lot of challenges across tissue integration beyond more data generation. Just the analysis itself is far from trivial. And so I'm going to read them quickly. So Shyam, I think you put this up, right? How can we identify tissue-specific cell states when sample prep, library prep protocols vary so much across tissue? When these factors are all confounded, how do you deal with this? How to decide, and I know the integration team has been thinking deeply about it, but how do you decide which computational methods and benchmark to use for integration across tissue? What type of modality and data set should we prioritize? When I say what type, here I'm talking again about modalities. Which computational parameters should be optimized, and what's ground truth? We're not going to solve this in 20 minutes, but just to point out, but just to point out that beyond just more data, like the actual process of integrating across tissue is far from trivial, beyond just collecting a lot of annotation metadata, and it probably needs to be an innovative track around this thematic. It's probably not what you want to say, but like you don't want to talk anymore. Okay, about this. And then there's the other two pieces, in case someone wants to comment. There's a validation piece of it, which I know there's a whole session around function tomorrow, so we can leave it aside. And then, importantly, how do you want to use that integrated atlas? What type of queries you're hoping to do? And that actually very much defines how we approach the integration, because it ties to what queries do we care about in the integration atlas. And it's probably the first question we should have put there, because that's going to define how we go about it. And some people are in agreement. Yes, go ahead.
Yeah, actually, I'm going to make a question about what I forgot her name, but just said that she's not in any bionetwork because she works across tissues. And that's kind of my position also. I work with modeling of cell types, so I don't care about one tissue specific, but like the whole picture. So and we have only 20 minutes to discuss this lot of things that we are all interested in. So as with the genetic diversity bionetwork, wouldn't there be room for a cross-tissue bionetwork? I mean, to kind of organize people and get, because perhaps we don't need just more data, we need more people and more time and more attention in a way. So I just want to raise the question, I mean, wouldn't there be room in HCA for a cross-tissue bionetwork?
It's a good question. I am not going to, if I answer as Chloe talking and not as other hats that I wear. It's a good question. I do think that it helps in dissecting the different issues we need to tackle. And it needs to be a community. So the short answer is yes, I don't know in which shape or form, but that community needs to be made of not only of folks that deeply care in terms of their biological expertise about a particular cell subset that is shared. We also need folks with computational expertise who can think more deeply about methods development, because these are not trivial. It's not something can start tomorrow morning. That's why we brought this up. They need to have the conversation together. And then, of course, we need those that still are generating data, because we need to understand, what type of proof of concepts we need, what type of data sets we need to benchmark this. And so, yeah, maybe you're right. I don't know that we call it a bionetwork, but it might be a working group. It's a good thought, and that's why we want to have this session, because this is the next frontier, right? Like, I think Sergio said, like, we're not talking tissue atlas, this whole body atlas. So how do we go there? What are the next steps? Yeah, you're right. Like, it's not in 20 minutes we're going to solve this. But there are big questions to tackle, and we need to figure out the minimal unit to kind of be able to move forward. And in that, there's not only data set identification, it's a lot of computational methods development. And it's, I don't know, it's not going to be simply ML framework who's going to solve this. At the very back, PJ, yeah.
Hi, I'm JP from KAIST, and I'm thinking a lot about the ground truth. And one thing that happens with the multiple data integration is, as the more and more data that we integrate, we lose a bit more of resolution. So we kind of end up with some kind of fuzzy atlas in the end. And most of the confounders is kind of different techniques. So I'm thinking maybe we can just focus on 2 or 3 main techniques, like 10x3 prime, 10x 5 prime, and maybe like combinatorial indexing, and then build up the ground truth for each of them. And in the end, we can compare the conclusion from each of the atlases, sub-atlases, and then just take the one that are robust across the modalities. So that's one thing that I've been always thinking about. So maybe you can try start building mega atlases, focusing on specific sort of techniques.
So I have, thank you. Keep that mic for a second. What can you define what you mean by ground truth? I think I mean, there's no right or wrong answer here. I mean, the problem is defining ground truth. I don't think we're defining it the same way. I know, different word. Yeah. I know. I did a funny play on word. I know.
I think ground truth is somewhere like a latent space from SSPI, and from different chemistry, we are just looking at them with a different prism. So if we just have 3 big atlases with the same technique, so that within itself it doesn't have a technical batch effect, and if we average it, I think we can just come closer to the ground truth.
See, that's interesting. That's why I want to. Yeah, I realized I made a funny play on word. I define my ground truth very differently as an immunologist and biologist. If I have defined new cell types and I've now defined my markers with these multi-omics and prospectively isolated the cells and isolated a lot of cells from different individuals and prospectively profile each is the same. Biologically, it's my ground truth, and then when I see, I think that's what Matas and other called seed annotation, and I want to see where it lands. That's my ground truth. It's like my ground truth is rooted in biology. But I think one of the challenges we're having is that across different communities here, we're not defining our ground truth the same way. And so while we're all having a conversation about ground truth, we're not talking always about the same thing. I know you have a question. I'm gonna Michaela, and then you two, but Michaela first.
So just talking about the ground truth, I think your approach would be good to define what we were talking about before, another type of core. But in my opinion, to get to the ground truth, that's where you need the orthogonal assays. You need a validation with different assays, which could be still gene expression, but then if you want to go a little bit further, you might want to look at proteins. I personally wouldn't consider the ground truth integrating single cell and then assuming that's correct, because the integration across organs is so complicated that there is so much background and unknown, and we're really at the moment guessing where we're right. And then only by validating we can somewhat get closer to the truth by comparing across tissues.
All right, I know they've been dying of jumping in. Sorry, I've been not ignoring you.
I was just going to, come back to one of the questions. How we've talked a lot about the problems, and I would like to make a sort of pitch for actually how far we've come, and also a pitch for diversity. So, first of all, yes, when we look at tissue, of course, the tissue prep confounds the data and so forth. But already we've moved from single cell to single nuclei where we don't have dissociation. So we already have a technology where the differences are a lot less than they were. We can multiplex across tissues so that the supereffects are the same. So there are actually things that we know how to address. And also the other thing is, like, just in the human lung cell atlas, which of course Malta led the integration of, there's already over 400 individuals. And that of course has a lot of women and a lot of men, and has people from across the age groups. And if this is being replicated across many of the different atlases, we can then start to pull out all the fibroblasts from the lung cell atlas and compare them to the fibroblasts from the heart cell atlas. And I really think we shouldn't try to build the one integrated atlas that will address all of our questions, but we should aim to generate data that is as interconvertible as we can make it at this point, and then really let people come up with their own biological questions and do the integrations they need to do to answer the questions they have.
Malta, you want to add to it? Malta, sorry, Shyam.
Malta can add to it too. I wanted to pick up on a point Jong-un made about over-integration. I think it's a very valuable point that's sometimes ignored. You just assume the more you integrate, the better your answer gets. But there is a sweet spot beyond that. If you're integrating things with massive batch effects, then you're actually blurring out the signal and your cell type definition actually gets worse. And yeah, so thanks. I presume that's a point you were making.
Okay,
yeah,
yes.
yeah.
Thank you for clarifying. Yes.
Thanks for making that point. And
go ahead, please.
Wanted to make another. Oh, okay. Well, I'll pass. Let other people speak. So,
we'll come back to you. There's a hand.
Hi, Stefan Ivanovic, S2 Genomics. We make a tissue dissociation system called the Singulator. So I totally agree with nuclei are better than cells. If you haven't seen the poster by Memorial Sloan Kettering, they compare different ways to make nuclei. And one of the things you'll see is that they don't all give the same answers. There's a Venn diagram, and the different methods give you different cell types or genes. So nuclei are better, they're not perfect. So one of the questions for the group is, are you keeping track of how did you make your cells or nuclei? Because that's really important, and that will change as we get better as manufacturers, as the community gets better. And then the last thing is on the ground truth. To me, the ground truth will partly be spatial, where you'll get to see what did the sample prep methods miss. It may not have as much detail, but you'll start seeing, oh, these cell types we're not seeing in cells. We're not seeing them in nuclei. So it'll be a component of the ground truth.
So you had a question among your comments, which is, are we keeping track? And so yes, there's different tiers of metadata for every data set that we are integrating. There's two main tiers. The first one is related to what you're saying, what we call tier 1. It's related to all the technical metadata about the sample collection, the sample processing, the protocols tied to it. We actually encouraged data contributors to even tie their protocols in protocol IO. That's really the extra step. But at the very least, we do have questions so we can track this, because we're well aware that digestion, warm, cold, nuclei, cell. I'm sure Martha has a lot of thoughts about the challenge of integrating nuclei and cell. All of this actually really matters. And so when we see an interesting batch effect, we want to be able to tie it to this metadata to try to understand if it's biology or actually technical. We also collect what we call it tier 2 metadata, which is more related to the biological system, the detailed and we also collect broad demographics, of course, but also detailed biology that needs to be tied to every biological network. Yes, I think Malta.
I think this question of ground truth is really interesting and really hard. When we kind of benchmark integration methods, we try to use different metrics to kind of assess what might be something that is related to ground truth. And in my mind, the real ground truth comes in the community annotation. So if you generate a version 1 of some kind of integration, and the community comes back and says, I agree, this is great. You know, if I can find my favorite cell type, that's great. That's not going to work everywhere. And so in my mind, for cross-tissue integration, you need wave 1 versions where there is at least community consensus on a subset of the cell types. You know, in the lung, we know we could have done better in immune cells, and hopefully we change that in next iteration. But once we have this kind of first version of a community consensus, then we can say, okay, now could we try to integrate with maybe novel methods or the same methods across these individual references, where we can treat that as a minimal ground truth at least, to then say, what are we destroying, or are we recovering more? And this is, I think, it brings to the point that was brought up also about over-integration. We need that before we go to the cross-tissue stuff.
Yeah. Like stepwise, yeah. I mean, Kerstin was related to it. Like we should be doing it per phase, and it's actually very important to be pragmatic about it, right? Like, so we can definitely, and we are actually, in case we didn't make it clear, tying it to the different waves of integration. Because couldn't agree more with Malta. Like that consensus annotation piece, we know that this consensus annotation will evolve with time. We'll put a timestamp to it because we know it's going to change as we add more samples and more modalities and increase diversity. And comparing to other tissue may also help us refine this identity. Is particularly relevant for immune cells. So yes, going stepwise and kind of learning along the way is really important, as opposed to just going whole body. But at the same time, in parallel, because we know that it's going to be so hard to do it at scale, I think it's not a bad thing to start investing a bit of resource. I don't know which resource yet. I'm not saying I have money. Just thinking maybe that's something we need to think about. Because it may be the same approaches, but maybe we need something very different. We have 5 minutes. Any burning comments you want us to take notes on? Or thoughts? Shyam, you want to speak?
No, I said they've been trying.
I've been ignoring them again? Oh, you want to?
Yeah, just a naive question. So you mentioned for the tier 2 you consider biological variations. So what exactly what biology do you consider? For example, do you consider circadian rhythm? Because, for example, some transcription factors, they vary drastically over the day. And I assume that when we sample these cells in a lab, we don't say that, right, this is the time of the day that we got this sample. And we don't consider it in a simple UMAP. So I was wondering, how do you incorporate these information in your integration?
I'm going to make one comment and then turn to Shyam in a second. And so the tier 2 metadata schema is an evolving schema. We started engaging, because of course there were schema from the get-go, but we have started engaging with every bionetwork because it's best to tailor the schema to every bionetwork, because there's actually specific needs for every organ system that needs to be where it needs to be tailored. Though I recognize that circadian rhythm is broad. And it's a bit tricky because we need to go with the metadata that our data contributors can provide, and this tends to be tied to their own study design, which is not always aligned with the full metadata schema that you may wish. And so it's important for us, for feasibility reasons, to establish tiers of priorities of what is a must across all data sets. It's especially important in the context of integration. So some of these technical metadata as well as broad demographics are a must. And then in terms of biological questions, this is where we highly encourage everyone to work with their respective biological network. And if you have not signed up to be part of a biological network, we highly encourage you to do so. There are actually QR code outside. You can scan your badge. You can be part of multiple biological networks. There's nothing tied to being part of a biological network. We just invite you to join our communities. And I didn't answer exactly your question about circadian rhythm. It is one of many fields that are part of the schema, but very few have collected it. There's a few data sets, and I think this is where I want to turn to Shyam for a minute, because they care very much about very detailed metadata schema so they can get to these questions. So there are some studies are designed for that, where we can learn from there and then figure out the gaps where we can expand. So before I get to the next person with a mic, do you want to add to this?
I was going to talk about something different, but yeah, we need as detailed metadata as we can get. But I think what we're finding is that for each network, the important metadata definition may be different. And so that's a conversation to be had within the bionetwork, I would say. Certainly for GDN, there's very specific metadata we need that may not be essential for the others. Yeah.
Before you expand on this, I just want to take a comment, and then we'll back to you. Just a comment at the back.
I mean, one aspect that is never taken into consideration is circadian rhythm in any of our tissue collection so far. That is something we really need to consider. There is a old story in Yuli Shibler's lab. He had a Canadian postdoc who used to work early in the morning. He found a transcription factor in the liver. somebody later on could not reproduce it because that guy was working in the evening and he harvested the tissue in the evening. So there are a lot of anecdotal evidence that people working at different time of the day had different set of data. So we need to consider that.
It's important, and yes, we're not saying this is not important. We're just saying that we need to, within the realm of every bionetwork and data contributors trying to define their key metadata, we encourage all of you to engage with your bionetwork, and I think you care about immune cells, so we can definitely coordinate. It's actually very difficult to, unless the study is starting, to get people to collect this information, but we can be very proactive moving forward. Okay, we have 1 minute and 17 seconds going. I want to give a chance to both Shyam and Kerstin.
In 1 minute, I'm going to raise a very difficult question, which I feel may not be answerable. Let's say we cross-tissue integration of T cells between blood and lung, for example. But the lung T cells come from one lab in the UK, say, that dissociated lung in one way and generated data one way. And the blood data come from Seoul National University, who collected PBMCs in one way. So most of the differentially expressed genes between the T cells in the lung and the T cells in the blood will be about differences in the sample collection process, sample dissociation process, and so on. How do we separate that from lung phenotype versus blood phenotype? So the two are perfectly confounded, right? The assay batch effect and the organ difference are completely confounded. At that point, is there anything we can do? And this is just a question. I don't have an answer.
Yeah, that was the question to answer. Oh, there are hands. Sergio. Oh, Malta.
More data. More batches. You average over. So.
Yeah, this is where it's important to think about the data sets. Sergio, you have some thoughts? And then Kerstin, I'll let you.
You can validate it. I mean,
yes, there's the whole
single cell is an in silico hypothesis generations, and then we validate it.
I think validated is a good answer everywhere, but here you'll just have so many artifacts. You already know that the validation will fail very often, right? When you have such massive.
Yeah, but they need you to function analysis or I don't know, whatever should help in this differentiation between batch or assay and biology.
Yeah, but I think it's a mix, right? Like more data, defining the right data sets for defining, I don't like the word ground truth in this case, but like some foundation. There needs to be a piece of validation. But yeah, this is why I say it's a really hard problem that we need to start thinking about right away as we're still generating data, because we need to plan accordingly so we have the right data sets to do this. Kerstin, any last words? So, you've been very positive, so you need to help us finish on a high note.
Yeah, so I would still come back to that, even though we all want to have this big atlas, at the end of the day, we all come with a biological question, and we have to have a good experimental design to address that biological question. And then if somebody, in these days, because you can do 1 million cells in a single experiment, it doesn't have to break the bank anymore. And if you then do find particular, let's go with the example of the circadian transcription factors, if you have a data set where you know exactly all your data set, you can then probably predict that back into this big atlas. So I think from my point of view, there's a lot to be said to have quite pure experiments and then use that to find hypotheses that you can then validate in a bigger integrated atlas.
Thank you. And so quick concluding notes. We've been taking, I should say not we, Lucia, I'm giving you credit. Lucia here, Robinson, has been taking great notes. We're going to be starting some of the key points. And I think it's important to circulate and try to get more thoughts so that we can think about how we prioritize this. I think it's very clear that this is not trivial. It's very clear it's stepwise. It's very clear we need to bring different members of the community to start thinking about this. It's not something we start one day and it happens. And it matters at every level, from the metadata, from the type of data we're generating, from the modalities we're generating, from the diversity, from the computational methods being done, from the current MVPs. And so this was tremendously helpful. Thank you for so actively participating and giving us your feedback. We're all around if you have more thoughts. Thank you.